In this video we're going to be looking at the main steps in drug discovery. How are drugs actually discovered and made? Well, there's a couple of things that we need drugs to be able to do. We need them to be safe. We don't want to kill people. We need them to be effective, that is, they do what we sell them as being doing, and they must be economical, must be affordable, and lastly they must be usable. A drug would be no good if you have to inject it every few minutes just to cure a headache. So what that means then is that we need to first of all find the steps that we need to go through are we need to find a target. So for example, typically a target might be a receptor in a cell, and then our drug might come along, bind to that receptor perhaps and cause it to open and cause an influx of, let's say, sodium ions, and then the result of that would be a cure. So you would need to understand your biological system to be able to figure out that we actually want to target this receptor here. So we find our target, and that's usually done using biological knowledge or sometimes new genomic approaches, for example. Once we've got a target, we need to find what we call a lead, which is we need to find this, which is the molecule which is actually going to hit the target. And so how can we do that? Well, we can screen large numbers of compounds until we find a hit. That is, a drug which we know opens our ion channel. The next thing we need to do is we need to optimize. So the idea here is, if I just spell this properly in the first place, optimize. The idea is that, great, we've got a drug, we found a molecule which has a particular action, and we find maybe it's got a particular shape. Now the thing is this molecule might give us a small response, but we want to find the best molecule that gives us a nice big response. So we might try, for example, adding a group here, or perhaps taking off a group down here and seeing if that makes it any better. So then we screen more compounds that are related to this one. Then we need to find out things like the pharmacokinetics, toxicity, speed of action, which is sort of under pharmacokinetics, I guess. Then we need to find out, is it okay in humans? Does it still work? Does it kill people? And then last of all, when we've done all this, we need regulatory approval. So this is basically the design of drug discovery. And we can break these steps down a little bit, because we refer to this bit here as the actual drug discovery, confusingly. And this part here are the clinical trials. And we have phase 1, phase 2 and phase 3, with more and more people involved as we go a bit later on. So we'd have a small number here of people in phase 1, we might have hundreds in phase 2, and maybe a few thousand in phase 3.